Renal mechanism for pH regulation. The role of kidneys in the maintenance of acid-base balance of the body is highly significant. The renal mechanism tries to provide a permanent solution to the acid-base disturbances. This is in contrast to temporary buffering system and short-term respiratory mechanism. The kidneys regulate the blood pH by maintaining the alkali reserve besides excreting or reabsorbing the acidic or basic substances as the situation demands. Urine pH normally lower than blood pH. The pH of urine is normally acidic. This clearly indicates that the kidneys have contributed to the acidification of urine. It is formed from the blood pH 7.4. In other words, the H plus ions generated in the body in the normal circumstances are eliminated by acidified urine. Hence, the pH of urine is normally acidic, that is approximately 6, while that of blood is alkaline 7.4. Urine pH, however, is variable and may range between 4.5 to 9.5, depending on the concentration of H plus ions. Carbonic anhydrase and renal regulation of pH. The enzyme carbonic anhydrase inhibited by acetazolamide is of central importance in renal regulation of pH, which occurs by following mechanism: excretion of H plus ions, reabsorption of bicarbonate, excretion of titrable acid, excretion of ammonium ions. Carbonic anhydrase and renal regulation of pH. The enzyme carbonic anhydrase inhibited by acetazolamide is of central importance in the renal regulation of pH, which occurs by following mechanisms: excretion of H plus ions, reabsorption of bicarbonate, excretion of titrable acids, excretion of ammonium ions. Urine pH normally lower than blood pH. The pH of urine is normally acidic. This clearly indicates that the kidneys have contributed to the acidification of urine. It is formed from the blood plasma pH 7.4. In other words, the H plus ions generated in the body in the normal circumstances are eliminated by acidified urine. Hence, the pH of urine is normally acidic, while that of blood is alkaline. Urine pH, however, is variable and may range between 4.5 to 9.5, depending on the concentration of H plus ions. Carbonic anhydrase and renal regulation of pH. The enzyme carbonic anhydrase inhibited by acetazolamide is of central importance in the renal regulation of pH, which occurs by following mechanisms: excretion of H plus ions, reabsorption of bicarbonate, excretion of titrable acid, excretion of ammonium ions. Carbonic anhydrase and renal regulation of pH. The enzyme carbonic anhydrase is of central importance in the renal regulation of pH, which occurs by 4 mechanisms: excretion of H plus ions, reabsorption of bicarbonate, excretion of titrable acids, excretion of ammonium ions. Excretion of H plus ions. Kidney is the only route through which the H plus can be eliminated from the body. H plus excretion occurs in the proximal convoluted tubules, renal tubular cells, and is coupled with regeneration of HCO3 minus bicarbonate ions. The process depicted in figure occurs. So, carbonic anhydrase and renal regulation of pH, they are closely related. The enzyme carbonic anhydrase is of central importance in renal regulation of pH, which occurs by following mechanisms: excretion of H plus ions, reabsorption of bicarbonate, excretion of titrable acids, 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 excretion of ammonium ions, excretion of H plus ions. Kidney is the only route through which the H plus can be eliminated from the body. Kidney is the only route through which H plus can be eliminated from the body. So, H plus excretion occurs in PCT and is coupled with regeneration of HCO3 minus. The process is depicted. See, in blood and renal tubular cell and tubular lumen is there. So, renal regulation of blood pH by excretion of H plus ions. So, in this, what happens is the sodium from the renal tubular cell, from the tubular lumen, it is sent to renal tubular cell, then sodium is sent to the blood. Okay, renal tubular cell, what happens? HCO3 minus is combines with H plus. What happens in renal tubular cell? Carbon dioxide and water, they combine to form by H2CO3 bicarbonate. H2CO3 bicarbonic anhydrase. Then this H2CO3 split into HCO3 minus and H plus. Then the HCO3 from renal tubular cell excreted into is reabsorbed into the blood. HCO3 minus and the H plus is sent into the tubular lumen, and this H plus will combine with B minus and HB will be excreted. Okay, so this happens in the renal regulation of blood. How H plus is excreted means the carbon dioxide and water, they combine with the help of carbonic anhydrase in renal tubular cell only, they combine in renal tubular cell. And then carbonic anhydrase converts H2 and CO2 into H2CO3 and HCO3 minus and H plus. Then HCO3 minus and H plus. The HCO3 is reabsorbed into the blood, and the H plus is sent into tubular lumen. This H plus combines with B minus and form HB, and this HB is excreted. This HB is excreted. And this HB is excreted. Tubular lumen sodium is sent into renal tubular sodium, and this is sent into blood. The carbonic anhydrase and renal regulation of pH. The enzyme carbonic anhydrase is inhibited by acetazolamide. It is of central importance in renal regulation of pH, which occurs by excretion of H plus ions, reabsorption of bicarbonate, excretion of titrable acid, excretion of ammonium ions. So this occurs here. Carbonic anhydrase and renal regulation of pH. The enzyme carbonic anhydrase, it can be inhibited by acetazolamide. The carbonic anhydrase enzyme is of very central importance in the renal regulation of pH because it combines the carbon dioxide and water and converts into bicarbonate H2CO3, carbonic acid H2CO3, and then it converts into bicarbonate in H plus, bicarbonate sent into blood, and H plus sent into tubular lumen where it is excreted. So this is the excretion, how excretion of H plus takes place. Now talking about carbonic anhydrase, it catalyzes the production of carbonic acid from carbon dioxide and water in renal tubular cell. This carbonic acid then dissociates it to H plus and HCO3 minus. H2CO3 dissociates into H plus and HCO3 minus. The H plus ions are secreted into the tubular lumen in exchange for sodium. Sodium ion in association with bicarbonate ion HCO3 minus is reabsorbed into the blood. The Na plus association with HCO3 minus is reabsorbed into the blood. This is an effective mechanism to eliminate acids from the body with the simultaneous generation of HCO3 minus. The latter adds up alkali reserve of the body. The H plus combines with the non-carbonate base and is excreted in urine. Then it is excreted in urine. The carbonic anhydrase catalyzes the production of carbonic acid from carbon dioxide and water in renal tubular cell. H2CO3 then dissociates it to H plus and HCO3 minus. The H plus ions are secreted into tubular lumen in exchange for Na plus. The Na plus in association with HCO3 minus is reabsorbed into the blood. This is an effective mechanism to eliminate acids from the body with the simultaneous generation of HCO3 minus. The latter adds up to the alkali reserve of the body. The H plus combines with the non-carbohydrate base and is excreted, non-carbonate base, and is excreted in urine. It is excreted in urine. Reabsorption of bicarbonate. Reabsorption of bicarbonate. Reabsorption of bicarbonate. This mechanism is primarily responsible to conserve the blood HCO3 minus with the simultaneous excretion of H plus ions. The normal urine is almost free from bicarbonates. So, reabsorption of bicarbonate. This mechanism is primarily responsible to conserve the blood bicarbonate. The simultaneous excretion of H plus. The normal urine is almost free from bicarbonate HCO3 minus. So what happens here is renal regulation of blood pH, reabsorption of bicarbonate. One is excretion of H plus, there is reabsorption of bicarbonate. So here you can see that in blood, renal tubular cell and tubular lumen. So in renal tubular cell, water and carbon dioxide combines to form carbonic acid H2CO3 bicarbonic anhydrous. And this H2CO3 converts into HCO3 minus and H plus. The HCO3 minus along with the sodium is eliminated from the renal tubular cell into the blood. It is sent. The HCO3 minus and H plus is excreted into tubular lumen where it combines with the okay. So how bicarbonate is regulated in the sense water and carbon dioxide they combine to form carbonic acid bicarbonic anhydrous, then bicarbonate and H plus is formed. The H plus is excreted into tubular lumen where from the plasma it combines with HCO3 minus and again it forms H2CO3 and then carbonic anhydrous, then bicarbonic because it can do reversible reactions also. H2CO3 carbonic acid is converted to carbon dioxide and water. This carbon dioxide and water is sent into the renal tubular cell where it forms the water and carbon dioxide they combine in the presence of carbonic anhydrous to form H2CO3 and the bicarbonate HCO3 minus is sent into blood along with sodium ions. The sodium from the tubular lumen enters into renal tubular cell. From there it is sent into the blood. From there it is sent into the blood. So the water and carbon dioxide they combine in the presence of carbonic acid to form H2CO3. This mechanism is primarily responsible to conserve the blood bicarbonate with the simultaneous excretion of H plus ions. The normal urine is almost free from HCO3 minus. This is explained. So bicarbonate freely diffuses from the plasma into tubular lumen. Bicarbonate freely diffuses from plasma into the tubular lumen. Here bicarbonate combines with H plus secreted by tubular cells to form H2CO3. H2CO3 is often cleaved by carbonic anhydrase to form carbon dioxide and water. As the carbon dioxide concentration builds up in the lumen, it diffuses into the tubular cells along with the concentration gradient. So bicarbonate freely diffuses from plasma into the tubular lumen. So here HCO3 minus combines with H plus secreted by tubular cells to form H2CO3. H2CO3 is then cleared by carbonic anhydrase of tubular cell membrane to form the carbon dioxide and water. As the carbon dioxide concentration builds up in the lumen, it diffuses into the tubular cell, into the renal tubular cell along the concentration gradient. In the tubular cell, carbon dioxide again combines with water to form H2CO3, which then dissociates into H plus and HCO3 minus. The H is secreted into the lumen in the exchange for sodium. The HCO3 minus is reabsorbed into plasma in association with sodium. In association with the sodium, HCO3 minus is reabsorbed into the plasma. Reabsorption of HCO3 minus is a cyclic process with the net excretion of H plus or generation of new HCO3 minus. So reabsorption of HCO3 minus is a cyclic process with the net excretion of H plus or generation of new HCO3 minus. This is because the H plus is derived from water. This is because the H plus is derived from water. This is because the H plus is derived from water. This mechanism helps to maintain the steady state and not be effective for the elimination of H plus or generation of new HCO3 minus. So this is because the H plus is derived from water. This is because the H plus is derived from water. This mechanism helps to maintain the steady state and will not be effective for the elimination of H plus or generation of new HCO3 minus. So this is not for elimination. This is just to maintain the steady state. This mechanism is to maintain the steady state. It is not for effective elimination of H plus or generation of new HCO3 minus. It's just for the maintenance of the steady state. Okay, excretion of titrable acid. Excretion of titrable acid. So, titrable acidity is a measure of acid excreted into urine by the kidney. This can be estimated by titrating urine back to the normal pH of blood. In quantitative terms, titrable acidity refers to the number of milliliters of N by 10 NaOH required to titrate 1 liter of urine to pH 7.4. Titrable acidity reflects the H plus ions excreted into urine, which resulted in a fall of pH from 7.4. So, excretion of titrable acid. Titrable acidity is a measure of acid excreted into urine by the kidneys. Is a measure of acid excreted into urine by the kidney. Is a measure of acid excreted into urine by the kidney. Excretion of titrable acid. Titrable acidity is a measure of acid excreted into urine by the kidney. Titrable acidity is a measure of acid excreted into urine by the kidney. Is a measure of acid excreted into urine by the kidneys. So, excretion of titrable acid. Titrable acidity is a measure of acid excreted into urine by the kidney. Titrable acidity is a measure of acid excreted into urine by the kidneys. It is a measure of acid excreted into urine by the kidney. Titrable acidity is a measure of acid excreted into urine by the kidney. Measure of acid excreted into urine by the kidney. How much acid is excreted into urine by the kidney? This can be estimated by titrating urine back to the normal pH of blood. Quantitative terms, titrable acidity refers to the number of milliliters of N by 10 NaOH required to titrate 1 liter of urine to pH 7.4. Titrable acidity is a measure of acid excreted into urine by the kidney. This can be estimated by titrating urine back to normal pH of blood. So, excretion of titrable acid is a measure of acid excreted into urine by the kidney. This can be estimated by titrating urine back to normal pH of blood, that is 7.4 in quantitative terms. Titrable acidity refers to the number of milliliters of N by 10 NaOH required to titrate 1 liter of urine to pH 7.4. So actually, titrable acidity is a measure of acid excreted into urine by the kidney. How can we estimate So this can be estimated by titrating urine back to normal pH of blood, that is 7.4. In quantitative terms, titrable acidity refers to the number of milliliters of N by 10 NaOH required to titrate 1 liter of urine to pH 7.4. Titrable acidity reflects the H plus ions excreted into urine. Acidity refers to the number of milliliters of N by 10 NaOH required to titrate 1 liter of urine to pH 7.4. A number of milliliters of N by 10 NaOH required to titrate 1 liter of urine to pH 7.4. Titrable acidity reflects the H plus ions excreted into urine, which resulted in a fall of pH from 7.4 of blood. Refers to the number of milliliters of N by 10 NaOH required to titrate 1 liter of urine to pH 7.4. So what is given here is refers to the number of milliliters of N by 10 NaOH required to titrate 1 liter of urine to pH 7.4. So 1 liter of urine, because urine pH is around 6, okay. So to convert it into 7.4 pH, how much milliliters of N by 10 NaOH is required to add? Titrable acidity reflects the H plus ions excreted into urine. Acidity reflects the H plus ions excreted into urine. H plus ions excreted into urine. H plus ions excreted into urine, which resulted in a fall of pH from 7.4. Titrable acidity reflects the H excreted into urine, resulted in a fall of pH from 7.4 phosphate. The latter in turn combines with hydrogen to produce the acid in which form the major quantity of titrable acid in urine is present. As the tubular fluid moves, oscillation of blood pH, excretion of titrable acid by phosphate buffer mechanism. So this is the blood, this is the renal tubular cell, and this is the tubular lumen. So we have water and carbon dioxide combining to form carbonic acid H2CO3 in the presence of carbonic anhydrase. Carbonic anhydrase is the fastest enzyme, and this enzyme is of central importance in renal regulation of pH, and this carbonic anhydrase can be inhibited by acetazolamide. It can be inhibited by acetazolamide. So carbonic anhydrase can be inhibited by acetazolamide. It is of central importance in renal regulation of pH. And renal regulation of pH occurs in 4 steps, 4 mechanisms: excretion of H+, reabsorption of bicarbonate, excretion of titrable acid, and excretion of ammonium ions. So talking about the excretion of titrable acid by phosphate buffer mechanism, in this renal tubular cell, blood and tubular lumen is there. So what happens? Carbon dioxide and water combines to form H2CO3, carbonic acid, in the presence of carbonic anhydrase. So then HCO3 minus and H plus. HCO3 minus is reabsorbed in the blood into HCO3 minus, and H plus into the tubular lumen. H plus is secreted into the tubular lumen. H plus is secreted into tubular lumen. Then with the exchange of sodium. So from where the sodium is coming into the tubular lumen? From where it is coming? So it is coming from the Na2HPO4. Okay, excretion of ammonium ions. This is another mechanism to buffer H plus ions tubular fluid. Other mechanism to buffer H plus ions secreted into the tubular fluid. The H plus ion combines with NH3 to form ammonium ions, H4+. The renal tubular cells deamidate glutamine to glutamate and NH3. This reaction is catalyzed by the enzyme glutaminase. The NH3 liberated in this reaction diffuses into the tubular lumen. It combines with H plus to form NH4+. Ammonium ions cannot diffuse back into the tubular cells and therefore are excreted into urine. So, talking about the excretion of ammonium ions. This is another mechanism to buffer H plus ions. So here what happens is H plus ions are secreted into the tubular fluid. The mechanism to buffer H plus ions secreted into the tubular fluid. The H plus ion combines with NH3 to form ammonium ion and H4 plus. The renal tubular cell deamidate glutamine to glutamate. The renal tubular cells deamidate glutamine to glutamate and ammonia and NH3. This reaction is catalyzed by the enzyme glutaminase. So the renal tubular cells deamidate glutamine to glutamate and ammonia. Glutamine to glutamate and ammonia. So glutamine is converted into glutamate and ammonia by the enzyme glutaminase. So the ammonia is liberated in this reaction. So this ammonia liberated in this reaction diffuses into tubular lumen, where it combines with the H plus to form an H4 plus. Ammonium ions cannot diffuse back into the tubular cells and therefore excreted into the urine. So the ammonium ions is a major urine acid. It is estimated that about half of 2/3 of body acid load is eliminated in the form of NH4 plus ions. For this reason, renal regulation by NH4 plus excretion is very effective to eliminate large quantities of acid produced in the body. This mechanism becomes predominant particularly in acidosis. So what happens here is renal regulation of blood pH, excretion of ammonium ions. So what happens here, the carbon dioxide and water, they combine to form carbonic acid H2CO3 by carbonic anhydrase. Then it will convert into bicarbonate HCO3 minus and H plus. H plus is secreted into tubular lumen, okay, in exchange with the sodium. Where the sodium comes from, we have seen the excretion of titrable acid by phosphate buffer mechanism. So the sodium comes from the disodium hydrogen phosphate. By giving it sodium, it will become sodium hydrogen phosphate, and then it takes H plus ions and it will become sodium dihydrogen phosphate. Okay. Okay, then this sodium this way, okay, renal tubular से glutaminase से glutamate और ammonia बन जाता. This ammonia enters into the tubular lumen, ammonia, and then it will convert into ammonium ion by taking H plus. And this ammonium ion cannot go back into the renal tubular cell, and hence it is excreted. So the glutamine, from where ammonia is coming, in the sense ammonia is coming from the glutamine, but the glutaminase converts glutamine into glutamate and ammonia. Carbon dioxide is the central molecule of pH regulation. So as is observed from the foregoing discussion, carbon dioxide is the central importance in the acid-base balance of the body. It has the ability to combine with water to form carbonic acid H2CO3, which can dissociate to bicarbonate HCO3 minus and H plus ions. A summary of the interaction between lungs, erythrocytes and kidney in handling carbon dioxide to maintain pH of the blood is depicted. The carbon dioxide generated by aerobic metabolism may be exhaled via lungs or converted to HCO3 minus ions by erythrocytes and kidneys to add up to alkali reserve of the body. So here you can see the carbon dioxide, it can be exhaled by the lungs or it can be taken up by the erythrocytes and transported, and HCO3 minus can be generated, or it can be taken up by the kidneys to form HCO3 minus, and H plus is lost in the urine by four mechanisms we have seen. Then HCO3 minus is taken up by the blood as the alkali reserve of the blood, of the plasma. The carbon dioxide is generated from the metabolism and reaches the blood as the carbon dioxide H2CO3, then can be eliminated into the lungs by exhalation. The carbon dioxide can be exhaled by the lungs or it can be managed by the erythrocytes and kidneys. How are erythrocytes managed? That we have already seen. Erythrocytes, they have hemoglobin. So this is all about the buffer system. And talking about buffers of intracellular fluids, the regulation of pH within the cell is as important as the discussed above for the extracellular fluid. The H plus ions generated in the cells are exchanged for sodium and potassium ions. This is particularly observed in skeletal muscle, which reduces the potential danger of H plus accumulation in the cell. Of pH within the cell is as important as that discussed above for the extracellular fluid. The H plus ions generated in the cells are exchanged for Na plus and K plus ions. This is particularly observed in skeletal muscle, which reduces the potential danger of H plus accumulation in the cell. So how intracellular it is maintained by the H plus ions generated exchange with sodium and potassium ions. Okay.